Thank you very much. It's a great pleasure to be here. I'm a scientist as much as for any other reason because of my high school chemistry teacher, so it's nice to be able to repay the favor. So imagine that it's October the 27th. That's the day after the Red Sox beat the Cubs in Game 7 of the World Series, about the 12th inning. And perhaps you had a little too much to drink. After all, it's hard to condense 85 years worth of celebration into one evening. And you wake up the next morning, the alarm clock is gaily trilling away. It's time to go see your young charges. And you struggle out of bed. It's dark, it's cold. And the first thing you try and do is put your socks on. And so there you are, struggling in the dark, fumbling. And one of the things that you will notice is that when you put your socks on, you always get your socks on all the way before you put your shoes on. And what I'm here to do this afternoon is to tell you how cells do the same thing. And you will notice that actually with this particular appropriately Bostonianly colored footwear, I don't actually have to wear socks. So it's not a requirement that I have socks on to be able to get the shoes to stick to my feet. But once I decide I'm going to try and put my socks on, however difficult the conditions are, and I gave you what I think is a worst case scenario, it will still be true that I will manage to get the heel of the sock over the heel of my foot before I put the shoe on. And you can do it in the dark, so it's not just by, you don't look at your foot. And so this is an example of a mechanism, a biological mechanism, to make sure that one thing gets finished before the next thing gets started. And the reason in this case is it would hurt like hell to wander around all day with your sock halfway up your foot and grinding into the bottom of your foot. Okay. So I want to start out by reminding you of something which I doubt any of you need reminding of. This is the structure of DNA as posited by Watson and Crick in 1953. This is the 50th anniversary. This is going to be my Halloween costume with my daughter. And this discovery instantly suggested how genetic information was encoded, particularly that because there are 4 different flavors or colors of these bases, the steps on the double helical staircase, it was obvious very early on that you could encode information simply by changing the sequence of these steps. And it turns out that that's true. And so here's a view that's slightly blown up. This is the diagram from the last slide, simply flipped over on its slide, and now scaled down and replicated many, many times. So in one of your chromosomes, there are on the order of 10 million helical turns like this. Okay? This is one gene's worth, something on the order of somewhere between 20 and a couple of thousand turns like that. And what that gene instructs your body to do is to make a particular protein. Okay, so this is a 3-dimensional picture of a protein. The individual atoms are these little balls. It is the particular shape of that protein and the chemical nature of the balls that are there that accounts for its ability to do some particular task. So that, for example, it could be, although this picture is actually not, it could be hemoglobin, the protein that is busily carrying oxygen from your lungs to all your other tissues as you sit here. Okay? So that's one way of looking at the gene. The gene is the information that codes for a particular protein. And here's another way. A gene is a particular part of a chromosome. And then again, the average chromosome in your body is between thousands and tens of thousands of genes long. And therefore, because the genetic information is carried on your chromosomes, and it is the genetic information that makes you you rather than your next door neighbor, that differentiates, let's say, Derek Jeter from Nomar Garcia-Parra, that it's very important for your body to be careful with its chromosomes. Okay? And what do I mean by that? So here are the relationship of chromosomes, cells, and peoples. This is a typical family. The mother, all of whose cells have 46 chromosomes, produces eggs which have half as many chromosomes by a specialized division called meiosis. The father, usually, at least in this family, the person with the lower IQ, produces, by the same sort of specialized division, sperms which have 23 chromosomes. So you've halved the number of chromosomes. The reason that you've halved them is when these come back together, they make an egg with 46 chromosomes, and it is the repeated division of that egg that makes the offspring of the British royal family, or you. It all just depends on who your parents are. And those repeated divisions produce about 10 trillion cells. You are a collective of about 10 trillion cells. And at least in evolutionary terms, the whole purpose of that collective is to make more eggs and more sperms so that your genes can be blasted off into the future. Okay? So this is a picture of the chromosomes that would be seen in one of your cells. There are two pairs, so that for each chromosome, you inherit one copy here from mom and one copy here from pop, one copy here from pop. There are 23 pairs, so there are 46 chromosomes. The only case where that's not true is for the X and Y chromosomes, where you can only inherit a Y chromosome from pop. Okay? And this is the picture of the chromosomes in a normal person's cell. This is someone who has Down syndrome. Okay? Normal, Downs. How many people have spotted the difference? Okay? For those of you who didn't, the difference is that a person with Down syndrome has an extra copy of this small chromosome 21. One of the things that's worth pointing out is you can have an extra copy of any chromosome, but with the exception of chromosome 21, the chance that a fetus will be born and be alive is essentially zero. So there's nothing special. You're no more likely to make mistakes with chromosome 21. It's just that because this chromosome is small and carries a relatively small number of genes, the imbalance in how many copies of each gene there are is small enough that individuals who are unfortunate to inherit this extra chromosome can survive, although they have a variety of different medical problems. Okay? Here's a second problem with cell division. So as I said before, your body is a community of 10 trillion cells. They have a common interest, producing eggs if you're a woman, sperm if you're a guy, introducing those eggs and sperms to each other if you're a teenager. And because all those cells are engaged in this communal effort, you have to make sure that cells don't misbehave and disrupt the community the way perhaps some of your charges do from time to time. And so normal cells obey extremely strict rules. They divide only when they're told to by signals that come from other cells. They would die rather than misbehave, so they're literally Victorian in their capacity for embarrassment, that if they do something that's against the rules and threatens that cellular community, they commit suicide. Okay? And they stay close to home, so it's rather like 19th century or even earlier society, where most people lived and died within 10 miles of where they were born. Okay? And they're very careful with their chromosomes. So that's Emily Dickinson. How many people know who this is, just out of interest? Sid Vicious, thank you very much. Okay, so there's at least one person who had some misspent years. Okay? So here's Sid Vicious, who is our analogy of a cancer cell. And these cells disobey the rules. They divide at will. When they behave badly, it doesn't kill them. That's actually an exception with the actual life of Sid Vicious. And they wander through the body, and they're extremely careless with their chromosomes. And it's this constellation of properties that makes cancer cells the enemy of this organized collective of cells that make up your body. And essentially, they are the rebels without a cause who are capable of destroying your body from within. Okay? And I just want to illustrate the difference between being careful with your chromosomes and being careless with your chromosomes. So this is the same picture I showed you. This is a normal cell, the Emily Dickinson type of cell, with 46 chromosomes. Emily Dickinson actually probably was not quite as pristine, but never mind. And this is a cancer cell, and what you will see is instead of there being 2 copies of chromosome 1, there are 4 copies of chromosome 1. If you look carefully, you can see they don't all look the same. These 2 have little bits that have been nibbled off the top. And these chromosomes here have part of chromosome 1 and part of some other chromosome. There's only one intact copy of chromosome 5. Okay? And this genetic instability is characteristic of all tumor cells. One of the things that's been appreciated in the last 15 to 20 years is that as well as tumor cells dividing at times and places where they ought not to, they are much less careful with their genetic dowry. And in fact, cancer is an evolutionary disease. These cells compete to replicate faster than the other cells in your body, okay? Because they can't give rise to eggs and sperm. This is a selfish rebellion, and one that's ultimately fruitless, because they kill the organism that could pass on genetic information. So what I want to do now is to walk you through in more detail and get you back to socks and shoes and checking on how things are done. I want to walk you through the process by which cells divide. So I'll start you here. This is a cell that's just been born. This is roughly what a typical human cell looks like. This blue part here is the nucleus, which contains the DNA and the chromosomes. As this cell ages, it grows bigger. And in a process that you can't see on my diagram, the chromosomes are duplicated. So where before there was one chromosome for mom and one chromosome for pop, for example, for chromosome 21, now there are 2 sister copies from mom and 2 sister copies from pop. Okay? And those are separated from each other in this process called cell division or mitosis. So what you see here is this, for example, is the 2 sister copies of chromosome 21 that came from pop. And they are lined up on this elaborate structure called the mitotic spindle, which I'll have more to say about in a second. And above them, for example, are the 2 copies from mom. And because they're sisters and they're identical, and you want to make daughter cells that are genetically identical from each other, you want one of the 2 copies to go this way, where it will get incorporated in this daughter cell. And you want the other copy, for example, from pop, to go in the other direction where it will be incorporated in this cell, so that those cells will be genetically identical to each other. And it's during the process of cancer, making cells that are genetically not identical with each other is one of the things that leads to this evolutionary progression where the behavior of the cells becomes worse and worse. And so I thought I would show you a movie of all this. So this is a cell. It's in mitosis. This is one particular chromosome right here. Okay? And what you're going to see is you're going to see this cell line its chromosomes up on the middle of that chromosome segregation machine I call the spindle. And then you'll see the sister copies pulled apart and moving to opposite cells. And in the middle, at the end, you'll see a furrow, a little bit like a purse string that's closing the top of a purse, but now instead of being at the top of the purse, it's in the middle, and it's pinching one cell into 2. So let us see, God willing. We're supposed, there we go, movie. Okay, and you see the chromosomes pull apart like that. And I think what I'll do is I'll let the movie, and you see the cell now pinching in half? This purse string coming in. And you see it again. Here are the chromosomes, they jiggle about, they get lined up, they get pulled apart, and the cell starts to divide. Okay, and we'll see if this one I don't think is going to actually play for us. Okay, it would have just been another example of the same thing. Okay. Mitosis takes roughly about an hour. So in that movie, which took perhaps 10 or 15 seconds, things had been speeded up 200 or 300 times. It's a good question. Okay. So what I showed you before was how you segregate your chromosomes right. This is an example of how you segregate your chromosomes wrong. Here are 2 sister chromosomes, let's say the ones from mom. They're attached in a sensible way. So when this cell divides, this one will go in this direction, this one will go in that direction, and each of these cells will have 1 copy of that chromosome. This chromosome is misbehaving. The specialized regions that attach it to these little red railroad tracks, which I'll have something more to say about in a second, both of those are attached to the same end of this structure. Okay? Both copies from pop. And as a result of that, when this cell divides, they're both going to go the same direction. This cell is going to have an extra chromosome, because it's going to have 2 copies of this chromosome here. And this cell is going to have a missing chromosome because when the purse string cuts this cell in half, there are no copies of this chromosome over on this side. And here's an example of how that can be important in tumor progression. So there's a hereditary form of childhood cancer called retinoblastoma, where cells start to divide inappropriately in the retina of your eye, which ultimately leads to children losing their eyes. And what happens is the individuals who are going to suffer that disease actually get born with one chromosome that carries a wild-type copy of that gene. So this is called RB, just short for retinoblastoma. And one copy that carries a mutant copy of the gene. A mutation is just a change in the sequence of those colored steps on the DNA. Okay. And if you get this sort of division, where both of the copies that have the good gene on it, the non-mutated form of the gene, go to the same end of the cell, this cell ends up with two copies of that chromosome. This cell is missing that chromosome, and because the chromosome you inherited from your other parent was unfortunate enough to carry a mutation, you can't make any of this important protein, and because you can't make any of that important protein, you start to divide in times and places where you should not. Okay? So this is a graphic example of how important it is to keep track of your chromosomes. Okay? And here is meiosis. So meiosis looks a little bit different from mitosis. I'm not going to belabor things too much, but now the gray chromosomes are two copies from mom. The white ones are two copies from pop. And what's unusual about meiosis is in the first meiotic division, instead of the sister chromosomes splitting from each other and going to opposite ends of the spindle, they stay together and they go to the same end of the spindle. Okay? So these two, which are mostly gray, end up going to this cell, and these two, which are mostly white, end up going to that cell, so that in meiosis you produce progeny cells that are not genetically identical to each other, which is why your children are not genetically identical to you and why you're not genetically identical to your siblings, even though you have the same parents. Okay? And the two chromosomes, the chromosomes from mom and the chromosomes that are from pop that are going to segregate away from each other, are held together because there's been a cutting and pasting event called recombination. It literally is cutting and pasting the DNA. Now, you have probably been taught, and you may well have taught your charges, that this cutting and pasting is important for promoting evolutionary diversity and is one of the reasons that sex was invented. If you actually talk to people who work on evolution, that's a deeply controversial assertion, which has not been proved to be true. However, it is unquestionably true that making this linkage, which you make by recombination, as I'll tell you more about in a bit, is absolutely essential for getting the chromosomes that go from mom to go to this end of the spindle and end up in this cell, and the ones that go from pop to end up in this cell here. Okay? And what happens in Down syndrome, at least in about 2/3 of the cases, which are the result of mistakes in production of the egg rather than production of the sperm, is these two chromosomes, the two copies from pop and the two copies from mom, instead of going to opposite ends of the spindle, go to the same end, they both go into this cell. This cell is going to do another division where the sisters separate from each other. And as a result of that, this egg is going to have two copies of chromosome 21, and when it gets fertilized by a normal sperm, it will make a child who has 47 chromosomes, because it has 3 copies of chromosome 21, and it will have the various problems that are associated with Down syndrome. Okay? So this is another example about why it's important to take care of your chromosome. So what I want to do now is to tell you what it is that in principle you need in order to make sure that your chromosomes are segregated properly. So you need some machinery that will segregate the chromosomes and break the linkage between them. Okay? So that's what these red railroad tracks are. You will need some specialized region of the chromosome, here represented in black, which attaches the chromosomes to the railroad tracks. You want to make sure that the two sisters point in opposite directions so that when one attaches to this end of the spindle, the other one is likely to attach to the other end of the spindle. And finally, you'd like some checking mechanism, something that makes sure that the cell has its socks on before its shoes, in this case, that it has lined its chromosomes up correctly before it gives the signal to separate them from each other. So I'm going to walk you through those different things. So the sisters, we believe, are bound to oppose each other because as they are copied from a single parent to make a pair of sisters, they're bound together. If you like, this is chromosomes in bondage. And these green molecules are a special kind of protein called cohesin. You don't need to worry about the name. But it's basically, the current belief is that this is a hoop that surrounds the two sisters, holds them together, and in particular holds them so that these black parts, which are called the kinetochores, again, you don't really need to worry about the name. This is the part of the chromosome that attaches it to the railroad track, that those two black parts from the two sisters point in opposite directions. Okay? And you need something that will move the chromosomes along the railroad tracks, and so lo and behold, you have proteins which are called motors, and they act exactly like a little tiny mechanical motor. They can walk along a railroad track, and so you have them attached to the specialized part of the chromosomes that are going to attach to the railroad tracks.
Dr. Murray, is that using ATP when it moves along?
Yeah, so these motors hydrolyze ATP to ADP and inorganic phosphate to move the chromosomes along the railroad tracks. Okay? And the railroad tracks, this is the one name that's worth remembering just because it's actually descriptive. These are called microtubules. They are little, thin, rigid, hollow tubes. But they're the analogy of railroad tracks. And so what happens is that the cell grows an array of these, and this is the thing down which the chromosomes are going to move. But all this is done in a way that's rather different from the way you would build a railroad. So imagine that this is Boston and this is New York, okay? And you wanted to build a railroad between them, which people did in the 19th century. You would send out a bunch of surveyors with appropriate monies to bribe the local officials along the way, and you would decide the route that optimized geography and the minimal payment of bribes to connect Boston and New York, because if you look at the way the railroad goes between Boston and New York, they must have done something like that, okay? And then, having surveyed the route, you would lay down tracks. The way cells do it is different and apparently bizarre. So what the cell does is as if they said, okay, get a bunch of guys together, lay down railroad tracks, pick any direction, lay them down in a straight line for 300 miles. If you haven't hit Boston, tear them up and let's try it again, okay? And that's what these microtubules do. They literally fish for the chromosomes. So it's actually a very good analogy that they are like someone casting out a lure and bringing it back, and the chromosomes are the fish. And so the way that happens is the microtubules are polymers. They're made up of many identical copies of the same protein. And hopefully when I push the button, what you will see is that they line up, and when they hit this specialized attachment site, the kinetochore, that stabilizes that microtubule. So if you hit Boston, you don't tear the tracks up again, right? If you got to Worcester instead, you go, yeah, not where I was intending to go, right? So what happens is cells go into mitosis or meiosis. The nucleus breaks down, and the microtubules do this crazy business of searching for the chromosomes. When they find the chromosomes, they stick to them. And eventually, if you wait long enough, we're all dressed up and ready to go. So the sisters are held together by these hoops. The locomotives are straining mightily to go down these railroad tracks, but they can't go anywhere because they're going in opposite directions. It's literally like hooking two locomotives up back to back and then trying to go down the railroad track. Not much happens except for some wheel spinning. And the railroad tracks are going to lead these chromosomes to 2 points, and eventually the purse string is going to come down the middle and divide this cell in two. And the way you go from being all dressed up and ready to go to actually going somewhere is there is a molecular pair of scissors which snips the green hoops. And now the two engines aren't opposing each other, and they chunder off down the railroad tracks and move the chromosomes to the ends of this structure. Okay? And the cell's going to divide. And what the spindle checkpoint, this checking mechanism that makes sure that the chromosomes are properly lined up, is it controls the activity of the scissors. So if the cell detects chromosomes that aren't lined up the right way, it puts the elastic band over the blades of the scissors. So if you were teaching grade school instead of high school, your charges couldn't be busy chopping each other's ears off. A problem I have with my young children. Okay. So one of the things that's worth pointing out is that there are two sorts Let me just go back. So what this means, if there's a problem, the checkpoint will say, don't start snipping anything because we want to wait and see if we can fix this problem. And what I want to point out to you in the next couple of minutes is that there are some problems that can be fixed just by waiting, and there are other problems that cannot. And here's an example of a problem that can be fixed by waiting. Here's a pair of sister chromosomes, and one of the two copies is attached to a microtubule, and the other one hasn't. So this hasn't yet been caught by any fisherman. If you just wait, and there's enough fishing going on, since this one is pointing more or less towards this other half of the cell, eventually you'll catch this attachment site. The pair of chromosomes will move to the center of the spindle. They'll be pointing in opposite directions. Everyone's ready for the molecular snip, snip, snip. All right? And what the chromosomes like this, which haven't bound to any microtubules, do is they send a chemical signal. They say, yo, we don't have any microtubules. Wait up. Okay? But there are also some problems that can't be fixed by waiting. And this is an example of such a problem. In this case, the two sisters have made a mistake, and even though they were supposed to point back to back, the linkage between them got a little floppy, and they ended up attaching to the same end of the spindle. Okay? And waiting isn't going to help here. Okay? They're already facing this way, so that it's hard for microtubules from over here to find them. And if this cell goes into division, both of these copies end up here, and as we talked about before, that's bad. Okay? And waiting's not going to help you. Okay? Because these chromosomes, they're already attached to microtubules. They're just attached to microtubules from the wrong places. That is, the two sisters are supposed to attach one to microtubules from over here, one to microtubules from over here, because that's going to pull them in opposite directions. And these guys got it wrong. Yeah.
You said that the chromosome sends a chemical signal.
Yep.
Is the signal that I am attached to my kinetochore, or is it a signal that I'm attached to the opposite pole? What's it? What?
Hold the question. We'll get there. Okay. It's an excellent question. Okay. And this goes back to looking at meiosis, where a lot of the experiments that I'm going to talk about in a second were done. And again, I'm reminding you that this is a pair of sisters from mom, this is a pair of sisters from pop. They're held together because there's been this cutting and pasting, this genetic recombination between them. And what we do in some of our experiments is we put little green dots on the chromosomes so we can see them float around inside a yeast cell. So we can actually look at individual chromosomes and see where they are. And that's to introduce you to actually my all-time favorite experiment done by a very talented biologist named Bruce Nicholas. And so he was interested in the question that you asked me, what is that signal? Okay? Is it whether you're attached or not, or is it something else? And so he did the following experiment, which requires extraordinary manual dexterity. So he looked at male grasshopper cells that are making sperm, so-called spermatocytes. And they're going through meiosis. Here is a spindle in meiosis 1. This is one pair of sisters. This is another pair of sisters. They're held together by recombination. He took a very, very fine glass needle, and he bangs. He goes bang, 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 bang. And eventually he can break this linkage between the chromosome and the microtubule. And having broken it, he can push this kinetochore over here, so it now attaches to this pole here. Okay? When it does that, the cells sit around like this for a while. These two kinetochores move towards this pole, this end of the spindle. And eventually one of them lets go. And when it lets go, it has two choices. It can screw up again, and it can attach back to this pole. But if it does that, it will chunter a little further, and then it will let go. And you just keep, this is a sort of futile cycle. Alternatively, this kinetochore can attach to a microtubule from over here, in which case you restore this original configuration, and everything is perfectly stable. And Nicholas scratched his head, and he went, gee, what's the difference? And he had a very clever idea. He said, well, okay, the difference here is these two guys are being pulled in opposite direction. If somewhere in that linkage between the chromosomes and the microtubule, there is a spring, that spring would be taut here, and it would be flaccid down here. And maybe that's what's going on. And so the ingenious way he tested that idea is he took his little needle here, And he hooked this pair of chromosomes here, and he yanked, okay, very gently, not exerting any more force than the microtubules actually themselves exert on the chromosome. And he held these guys under tension. And as long as he held them under tension, this orientation was stable. So that shows that the signal, that if we go back to answer your question, the signal that these chromosomes are sending is, I'm not taught, and I'd like to be taught, please. T-A-U-T, not G-H. Okay? So now I want to finish up by telling you, walking you through how you can study this process in a simple organism, which is a lot less complicated than grasshoppers. And that's our friend, the brewer's yeast. So this is the yeast that's responsible for making the beer you drink, the bread you eat. And it can grow both as a haploid and as a diploid. And when you starve diploid cells in the appropriate way, they go through meiosis, just the way that the cells that make your sperm and egg go through meiosis. And they make 4 haploid products. And here is a micrograph of a yeast cell. This is a cell that's dividing. And this is one nucleus here stained with a dye that binds DNA. And this is another nucleus over here. And this is an organism. This is probably the best organism that exists at the moment for trying to understand the basic processes of cell biology, how cells keep track of their chromosomes, how they send some parts of the protein, some proteins to some parts of the cell, other proteins to other parts of the cell. It's just an organism where, partly because God smiled on us and there are certain things that people had no idea would be true turned out to be true, and also because a lot of people have worked on it, this is sort of the ultimate Rolls Royce of doing experiments in cell biology. Okay? And we use, as I mentioned before, we use jellyfish to see in the dark. So if you've been down on the Cape or to Rhode Island in the summer and you've swum at night, you will notice that the water turns this fantastic green glow. Okay? And people refer to that as phosphorescence, which is unfortunate because it's not actually phosphorescence, it's fluorescence. And what happens is that the organisms, dinoflagellates, some species of jellyfish, emit light. And that light, which is actually blue to start with, hits a protein and comes out as green light, okay? Which is what gives rise to the green glow. And so what people did about 10 years ago now was to identify the protein that is a little green light bulb. And what Aaron Straight, who was a graduate student who worked with me in collaboration with a guy called Andy Belmont at the University of Illinois, is they exploited that, which is a relatively recent advance, with an advance that actually got made in this very building by a guy called Wally Gilbert. And he is the guy who identified a protein called the lac repressor. So if you know about Jacob and Monod, they are the 2 French guys who discovered the principles of genetic regulation, and they posited the existence of a molecule called a repressor, which would control the expression of certain genes. And what Wally Gilbert did was to identify that molecule and the piece of DNA it bound to. So you can get lots of copies of that piece of DNA. This little black bulb binds very tightly to these little gray rectangles. And if you use the techniques of genetic engineering to attach the little green light bulb to the little black ball, you can make a whole series of light bulbs sat down on a piece of DNA, and you can look at the cell, and you can see the light. Okay? So this is what's called an ascus. This is the bag in which a yeast cell underwent meiosis, and it made 4 spores. 1, 2, 3, 4. Just the way gentlemen in the audience make 4 sperm. When women make eggs, they only make 1 egg from each division. Okay? And each one of those spores has a little green dot in it, and that little green dot is this piece of DNA. Through the marvels of genetic engineering, we can put that piece of DNA on whichever chromosome we want. We can put it at whatever position on the chromosome we would like to place it. And then we can follow it around. Okay? And this is Marion Schoen, who did the experiments that I'm going to show you when she was a graduate student. And so this is what meiosis looks like when it's done right. Okay? So here are little green dots we use to follow things. Mom, pop, first division. The kinetochores from mom and pop stay together. So this is different from all the other divisions. The second division looks just like mitosis. The sister kinetochores separate from each other, and you make 4 meiotic progeny. They each have 1 copy of this chromosome. Everything's cool. Wrong. So this is what happens when you do things wrong. Now the two pairs of chromosomes are attached to the same pole of the spindle. They both go to the same end at the first meiotic division. They line up in the second meiotic division so that you're going to separate these sister kinetochores. The gray ones are going to separate here. The white ones are going to separate here. You make two cells that have an extra chromosome, and you make two cells that have missing chromosomes. Okay? This is humans. These progeny can go on to give rise to Down syndrome. And so what we wanted to ask is, does the spindle checkpoint have anything to do with the accuracy of this process? And in particular, coming back to your question about what different ways you can tell things are screwed up, does it know that these chromosomes are unhappy? And the answer appears to be yes. So here is, so we've looked at two sorts of cells. We've looked at wild-type cells, and we've looked at mutants in the spindle checkpoint. We use various sorts of jiggery-pokery to isolate mutations in the spindle checkpoint. And if people want to ask questions, I can tell you how you do things like that. Okay? In wild-type cells, and so these again are the four spores. You can see a dot here in this cell, a dot here in this cell, a dot here in this cell. And this one is very hard to see, but I swear on my father's grave that it is there. Okay? And about 2% of the cells look like this, where you have two cells with no dots and two cells with two dots. That's wrong. Okay? And if you look at the ratio in the checkpoint mutant instead of wild type, 81% of them look the way they're supposed to, 19% of them look unhappy. Okay? So that's a tenfold increase in the rate at which you make mistakes when you eliminate the checkpoint. And so one of the questions that Marion wanted to ask, remember I said there's some sorts of problems that you can fix just by waiting if the chromosomes aren't attached to microtubules. There are other problems where if you have both chromosomes attached to the same end of the spindle, you can't fix the problem just by waiting. And she wanted to ask, is the checkpoint involved in that second sort of thing, the thing where you have to let go of something before you can line it up right? And so the argument is, maybe what happens is some of the time the chromosomes line up like this. Then the spindle checkpoint is actually doing that thing that happened in Bruce Nicholas's experiment. It's somehow stimulating one of these two kinetochores to let go of their microtubule, giving a chance to line things up properly. And in principle, we can look at that by putting green dots on the chromosome, because in this situation the dots are really close together because the chromosomes aren't being pulled apart. And here they're being pulled further apart, and we can look down the microscope and we can measure the distance between the dots. And so the results of Marion's experiment is she said, okay, I'm either going to say that the two dots were very close together or they were pretty far apart. In particular, they were far enough apart that I could be sure that I saw two distinct dots. And I'm going to look at the frequency of those different arrangements. So here's wild type. 2% of them have the dots close together, which we interpret, and interpret is an important word here, as meaning that the two pairs of chromosomes got attached to the same spindle. The sort of delay you can't fix just by waiting. And when you look at the mad mutant, when you look at wild type, 2% of them look like that, 98% of them look like this, which we interpret as the two chromosomes having attached to opposite poles of the spindle, which is what we want. And in the checkpoint mutant, so MAD2 is just the name of one of the genes that makes up this checkpoint, 24% of the time they look like this, and only 76% of the time they look like this. So we argue from experiments like this that the checkpoint is actually involved in fixing the defects where waiting won't help. And in particular, with regard to Down syndrome, we argue that the checkpoint does two different things. Here are a pair of chromosomes that are in trouble. There are two things we need to do to make sure that those do not give rise to an egg that is going to produce a child with Down syndrome. The first is we need to make damn sure that this cell doesn't go through cell division and segregate both of these chromosomes to the same pole. So we have to say to the scissors, wait up, no cutting. Okay? The other thing we need to do is we need to persuade this pair of chromosomes that are attached to one pole of the spindle that actually they're supposed to be attached, so that one pair of sisters is attached to this end and the other pair is attached to the other end. Okay? And we argue that the checkpoint does that. So what you could see from this is that as the checkpoint gets worse, it's sort of a double whammy. It makes it harder to delay division in cells that are going to do things wrong, and it makes it harder to correct errors in cells where the initial attachment of the chromosomes has been incorrect. And So can we relate this to the fact that Down syndrome, the incidence of Down syndrome rises very sharply with age? So this is a graph of the risk of having an infant with Down syndrome, if you're a woman. And this is the mother's age. And what you can see is that around 35, this risk gets to be about 1 in 200. And that's when the medical profession says, okay, the risks from amniocentesis, the risk of aborting a pregnancy through amniocentesis, which is when you go in and take some cells out and actually look at how many chromosomes there are, become less than the risk of having Down syndrome, and therefore it's reasonable to start doing amnio on mothers over 35. Okay? It's an interesting question that you could talk to your kids about. That's when the numerical risk gets to be the same. Those risks qualitatively are a little bit different. One's an aborted fetus, another's having a child with Down syndrome, and it's sort of an interesting thing for high school students to think about, well, okay, how commensurable are those risks with each other? Okay? And the interesting question is, why does this incidence rise like this? One of the things we know is that chromosome linkage is different in Down syndrome and normal infants. So if you look at where recombination occurs, where the cutting and pasting that happened between the maternal and paternal copies of chromosome 21 went on, it can either be very close to the kinetochores like this, or it can be very much up towards the tips of the chromosomes. And what's true about infants who have Down syndrome, and you can use genetic markers to show this is true, this sort of recombination is rarer in Down syndrome, and this is more common. So how could that be important? Well, the answer is, if the two chromosomes are hooked together, and now my elbows are going to be the kinetochores. If you tie my elbows together, maybe they're forced to point back to back, and so if I attach this elbow over to this wall, this elbow here has to attach to the other wall. Alternatively, if the recombination event is far away, like it is here, now it means that they're attached up near my fingers, and my two elbows can sort of flop around with regard to each other because there's some flexibility in this linkage. And now it's a lot easier to, get these two elbows attached to this wall. Okay? So we could go, okay, so that's a reasonable idea. But, and it's a big but, all the recombination has occurred by the time a mother herself is born. So a human female is born with all the oocytes that will give rise to eggs already produced. So all that happens is that once you get to puberty, and between puberty and menopause, every month, every lunar month, 1 or 2 of those oocytes matures and becomes an egg. So all the recombination has already happened. So it can't be true that this sort of recombination is common early in life and this sort of recombination is common late in life, because it ain't that way. Recombination